Good morning, everyone. My name is Helena Cerutti, and I am a biotechnology researcher of the R&D team of Diesse Diagnostica Senese. Diesse is an Italian company with integrated and entirely in-house production of in vitro diagnostic systems. Its headquarters are in Siena. Since its foundation in 1980, the company has developed, produced, and marketed innovative diagnostic systems, primarily in the field of immunology and ESR. The company has 3 production sites and a research center where new tests and new automated diagnostic tools are designed and implemented. At the beginning of 2020, when coronavirus infections began to affect Europe and Italy, we began to work intensively to develop highly sensitive and specific serological tests. At the end of the project, we decided to publish the work done. Our article is entitled Large-Scale Production and Characterization of SARS-CoV-2 whole antigen for serological test development. Considering the evolution of the worldwide pandemic, an improved insight into the development of specific antibodies after SARS-CoV-2 infection is of great importance. Actually, it is still unclear whether patients previously diagnosed with COVID develop immunity and whether this immunity remains. Therefore, serological investigations play an essential role not only to better understand the host response to the virus and provide a more accurate estimation of the spread within the population, but they also have an impact on vaccine and therapy development. In addition, a differential diagnosis between SARS-CoV-2 infection and other viral ones is essential. The aim of our work is to produce inactivated SARS-CoV-2 virus, to characterize it, and to use it in the development of serological tests. Cultivation of the virus in our BSL-3 laboratories has allowed us to file a proprietary patent protocol, thanks to the collaboration with a consultant from the Siena vaccine district and expert in the development of viral vaccines. For the virus cultivation, we used the Vero E6 cells. Vero E6 cells were cultured to a confluence monolayer and infected with the SARS-CoV-2 strain isolated for the first time in Italy at the National Institute for Infectious Diseases Lazzaro Spallanzani in Rome. The virus was cultured in a DMEM medium supplemented with 5% FBS to a low specific MOI of 0.05 and incubated at 37 degrees Celsius for 46 to 50 hours in roller bottles. Vero E6 cells were seated at 37 degrees Celsius in 24 well plates 24 to 72 hours prior to infection with the SARS-CoV-2 virus, and then further incubated. After the absorption step, 1 ml of DMEM supplemented with 5% FBS was added and the plates were re-incubated. The cytopathic effect was evaluated under the microscope every 24 hours by identifying cell rounding and detachment in monolayer cells. After 48 hours, CPE was evident. So, viral titers were calculated according to the Reed and Munch method, and the median infections dose per tissue culture was also determined. At 72 hours, the CPE is complete. As you can see in the image of the right, the cytopathic effect is even clearer and more visible using the plaque test. A protocol using beta-propionlactone, a commonly used reagent for viral inactivation in vaccine preparation, has been adopted and optimized for virus inactivation. After virus thawing, the BPL was added in 3 consecutive additions to ensure total inactivation, although it was already evident after the first addition. Viral inactivation was verified by SpA test. Each batch of inactivated virus was concentrated and purified by centrifugation. Batches were centrifuged to remove cell debris and concentrated by ultracentrifugation. The pellets were ultrasonically broken down and collected in a phosphate buffered saline solution. Batches were stored at -80 degrees Celsius. After thawing and before each use, protein levels in the virus stock were quantified using the Bradford assay. Each batch of viral antigen produced were quantified with SDS page and Western blotting analysis. In this study, SDS page was used to analyze 2 different batches of viral antigen, with and without fetal serum, to better analyze the structural proteins present in the SARS-CoV-2 virus. The batch containing fetal serum, number 1, was 671 mg/liter concentrated, while the serum-free batch concentration, number 2, was 178 mg/liter. SDS page results showed that the composition of the structural proteins were similar in both produced batches and contained the 4 structural proteins as expected. The spike glycoprotein band is just below 150 kilodalton. The band corresponding to the nucleic acid protein is below 50 kilodalton. The membrane protein is visible between 20 and 25 kilodalton, and the envelope is the lowest molecular weight protein, below 10 kilodalton. Subsequent investigation in Western blotting and ELISA were carried out with sera of convalescent patients after a confirmed SARS-CoV-2 infection, and sera from healthy donor collected prior to COVID-19 pandemic. Both analyses were concurrently performed with recombinant maps to verify the findings. In all panels, the first lane represents the standard molecular weight marker. We analyzed 10 serum samples, 5 positive and 5 negative, after electrophoresis under reducing and non-reducing condition. Western blotting of the convalescent serum samples showed a varying immune response between patients, and reducing and non-reducing conditions appeared to have a considerable effect on the response toward the N and S proteins. Reducing conditions displayed only a band corresponding to the N protein, supporting the hypothesis that the immunodominant epitopes of the S protein are mainly conformational rather than linear. Only 1 serum among those analyzed appeared to recognize the N protein. Presence of IgA and IgM were also detected. Similarly, the analysis with omercalmab, specifically targeting the N and stability 1 of the S protein, demonstrated the same difference between reducing and non-reducing electrophoresis conditions as those observed with the serum samples. Similarly, the sera and commercial maps were tested with the ELISA method and were found to have the same results as those obtained by Western blotting. Values are expressed in absorbance and index. Index is determined as a cutoff calculated from an arbitrary value of the average of the negative sera plus 3 standard deviation. In conclusion, we can affirm that the NATs remain the gold standard for early infection diagnosis, but the serological assay play a fundamental role in providing epidemiological analysis and enhance the understanding of immunity profile from recovered patients, serological therapies, and vaccines. In particular, Diesse developed serological diagnostic kits to rapidly detect the specific antibodies, IgM, IgG, and IgA, by using an indirect ELISA based on the crude extract from SARS-CoV-2 whole virus. Diesse Diagnostica Senese started by directly culturing the virus in mammalian cells. Our analysis showed that the culturing, inactivation, and concentration method used are adequate for whole virus production, since all of the structural protein were present with their native conformations preserved. Results from the Western blotting analysis identified that the nucleocapsid and the spike were the most immunogenic proteins, and sera from infected patients contained antibodies able to recognize both conformational and linear epitopes for the N protein, but only antibodies against conformational epitopes for the spike. Extensive experimentation has been conducted following this first analysis to confirm these findings, and 3 diagnostic kits for IgG and IgM and IgA have been approved for commercial use, and the CE marking was obtained on April 23, 2020. A large Diesse test panel is currently available for the diagnosis of SARS-CoV-2 infection. In addition to the 3 kits based on the manual ELISA method, we have also developed the kits applied on the Diesse Chorus instrument. Chorus are immunometry-based instruments on ready-to-use and simple determination diagnostic device. We have therefore available Chorus kits for the determination of total and specific antibodies for SARS-CoV-2. We also have the Antigen kit for the detection of the virus in the nasopharyngeal swab, and the neutralizing kit as a tool for evaluation of the post-infection or vaccine immune response. Thank you for your attention.